buffers, acid-base balance, and the diseases related to acid-base balance. The normal pH of blood is maintained in the narrow range of 7.35 to 7.45, that is slightly alkaline. Maintenance of blood pH is an important homeostatic mechanism of the body. In normal circumstances, the regulation is so effective that the blood pH varies very little. Changes in blood pH will alter the intracellular pH, which in turn influence the metabolism, example, distortion in protein structure, enzymatic activity, etc. It is estimated that the blood pH compatible to life is 6.8 to 7.8. Production of acids by the body. The metabolism of the body is accompanied by an overall production of acids. These include the volatile acids like carbonic acid or non-volatile acids such as lactic acid, sulfuric acid, phosphoric acid, etc. Carbonic acid is formed from the metabolic product carbon dioxide. Lactic acid is produced in anaerobic metabolism. Sulfuric acid is generated from proteins. Phosphoric acid is derived from organic phosphates, example phospholipids. All these acids add up the H plus ions to the blood. A diet rich in animal proteins results in more acid production by the body that ultimately leads to the excretion of urine, which is profoundly acidic. Production of bases by the body. The formation of basic compounds in the body in the normal circumstances is negligible. Some amount of bicarbonate is generated from the organic acids such as lactate and citrate. A vegetarian diet has a tendency for a net production of bases. This is due to the fact that vegetarian diet produces salts of organic acids such as sodium lactate, which can utilize H plus ions produced in the body. For this reason, vegetarian diet has an alkalizing effect on the body. Blood buffers. The body has developed 3 lines of defense to regulate the body's acid-base balance and maintain the blood pH around 7.4. The first one is blood buffers. It forms the first lines of defense. The second line of defense is respiratory regulation, and the third line of defense is renal regulation. Blood buffers. Buffers are solutions which can resist changes in pH when an acid or alkali is added. The composition of a buffer solution are mixture of weak acids with their salt with a strong base, or mixture of weak bases with their salt with a strong acid. A few examples are given below. Bicarbonate buffer, which is composed of carbonic acid and sodium bicarbonate. Acetate buffer, composed of acetic acid and sodium acetate. And phosphate buffer. Mechanism of action of buffer. Added H plus ions from a strong acid combined with A from the salt component of buffer to form the weakly dissociable weak acid, so that pH does not become as acid as it would be in the absence of buffer. Second case, added OH minus ions from the strong base combined with H plus ions derived from the HA, the weak acid, and form the weakly dissociable water molecule. Hence, the pH does not become as alkaline as would happen in the absence of a buffer. Renal mechanism of pH regulation. The role of kidneys in the maintenance of acid-base balance of the body is highly significant. The renal mechanism tries to provide a permanent solution to the acid-base disturbances. This is in contrast to the temporary buffering system and a short-term respiratory mechanism. The kidneys regulate the blood pH by maintaining the alkali reserve, besides excreting or reabsorbing the acid or basic substances as the situation demands. The enzyme carbonic anhydrase is of central importance in the renal regulation of pH, which occurs by the following mechanisms. The first one, excretion of H plus ions. Second one, reabsorption of bicarbonate. Third one, excretion of titratable acid. Fourth one, excretion of ammonium ions. First one, excretion of H plus ions. Kidney is the only route through which the H plus ions can be eliminated from the body. H plus ions excretion occurs in the proximal convoluted tubule and is coupled with the regeneration of bicarbonate. Carbonic anhydrase catalyzes the production of carbonic acid from carbon dioxide and water in the renal tubular cell. The carbonic acid then dissociates into H plus and bicarbonate. The H plus ions are secreted into the tubular lumen in exchange of sodium. The sodium in association with the bicarbonate is reabsorbed into the blood. This is an effective mechanism to eliminate H plus ions from the body by the simultaneous generation of bicarbonate. The latter adds up to the alkali reserve of the body. The H plus ions combines with the non-carbonate base and is excreted in the urine. Second one, reabsorption of bicarbonate. This mechanism is primarily responsible to conserve the blood bicarbonate with the simultaneous excretion of H plus ions. The normal urine is almost free from bicarbonate. Bicarbonate freely diffuses from the plasma into the tubular lumen. Here, the bicarbonate combines with H plus ions secreted by the tubular cells to form carbonic acid. Carbonic acid is then cleaved by carbonic anhydrase to form carbon dioxide and water. As the carbon dioxide concentration builds up in the lumen, it diffuses into the tubular cells along the concentration gradient. In the tubular cells, the carbon dioxide again combined with water to form the carbonic acid, which then dissociates into H plus ions and bicarbonate. The H plus ions secreted into the lumen in exchange of sodium. The bicarbonate is reabsorbed into the plasma in association with sodium. Reabsorption of bicarbonate is a cyclic process with the net excretion of H plus or generation of bicarbonate. This is because the H plus ions is derived from water. This mechanism helps to maintain the steady state and will not be effective for the elimination of H plus or generation of new bicarbonate. Third one, excretion of titratable acid. Titratable acid is a measure of acid excreted into urine by the kidney. This can be estimated by titrating urine back into the normal pH of blood. In quantitative terms, titratable acidity refers to the number of milliliters of N/10 sodium hydroxide required to titrate 1 liter of urine to pH 7.4. Titratable acidity reflects the H plus ions excreted into the urine, which results in the fall of pH from 7.4, that of blood. As already discussed, H plus ion is secreted into the tubular lumen in exchange of sodium ions. This sodium is obtained from the base, disodium hydrogen phosphate. The latter is in turn combines with the H plus ions to produce the acid sodium dihydrogen phosphate, in which form the major quantity of titratable acid in the urine is present. As the tubular fluid moves down the renal tubules, more and more H plus ions are added, resulting in the acidification of urine. This causes a fall in the pH of urine to as low as 4.5. Any further fall in the pH will cause depletion of sodium ions. Fourth one, excretion of ammonium ions. This is another mechanism to buffer H plus ions secreted into the tubular fluid. The H plus ions combine with the ammonia to form ammonium ion. The renal tubular cells deaminate glutamine to glutamate and ammonia. This reaction is catalyzed by the enzyme glutaminase. The ammonia liberated in this reaction diffuses into the tubular lumen, where it is combined with H plus ions to form ammonium ion. Ammonium ion cannot diffuse back into the tubular cells and therefore are excreted into the urine. Ammonia ion is a major urine acid. It is estimated that about half to two-thirds of the body acid load is eliminated in the form of ammonium ions. For this reason, renal regulation via ammonium excretion is very effective to eliminate large quantities of acid produced in the body. This mechanism becomes predominant, particularly in acidosis. Disorders of acid-base balance. The body has developed an efficient system for the maintenance of acid-base equilibrium, with the result that the pH of the blood is almost constant, that is 7.4. The acid-base disorders are mainly classified as acidosis, a decline in blood pH, and this can be either metabolic acidosis or respiratory acidosis. Metabolic acidosis is due to decrease in bicarbonate, whereas respiratory acidosis is due to an increase in carbonic acid. Alkalosis. There's a rise in blood pH. Alkalosis can be metabolic alkalosis or respiratory alkalosis. In case of metabolic alkalosis, there is increase in bicarbonate. In case of respiratory alkalosis, there's decrease in carbonic acid. These are clinical disorders owing to disturbances of acid-base balance. Acidosis, it is a fall in the bicarbonate carbonic acid ratio of the blood, which with a consequent lowering of blood pH accompanied by hyperkalemia due to potassium outflow from the cells and potassium retention from urine. Acidosis, as mentioned earlier, can be respiratory acidosis or metabolic acidosis. Respiratory acidosis is produced by pulmonary hyperventilation, as in asthma, respiratory paralysis, or lobar pneumonia. It is characterized by carbon dioxide retention in the blood. Metabolic acidosis, caused by excessive loss of bases in diabetic ketoacidosis, severe diarrhea, nephrotic acidosis, and hypoxic lactic acidosis. It is characterized by a fall in the plasma bicarbonate concentration, thus lowers the bicarbonate by carbonic acid ratio and the pH of the blood. Alkalosis. It is a rise in the bicarbonate carbonic acid ratio of blood with a consequent rise in the blood pH accompanied by a fall in serum potassium, hypokalemia, due to potassium influx into the cells and potassium depletion through urine. Alkalosis, as mentioned earlier, can be of two types: respiratory alkalosis and metabolic alkalosis. Respiratory alkalosis is caused by pulmonary hyperventilation in thermal panting, fever, mountain sickness, hysterical overbreathing, and voluntary hypopnea. It is characterized by an excessive removal of carbon dioxide from blood and consequent lowering of blood carbonic acid concentration. Thus, rises the bicarbonate by carbonic acid ratio in the blood, leading to rise in blood pH. Metabolic alkalosis arises from a non-respiratory factor such as violent vomiting, pyloric stenosis. It is primarily characterized by rise in plasma bicarbonate, thus rises the ratio of bicarbonate buffer pair and thereby increase the blood pH. Thank you.